Thank you very much, and it's a great honor to be asked to speak here, and luckily on my favorite subject, which is immunology, and I please stay in your seats because I know that many people are still traumatized by the first immunology lecture they ever heard, and hopefully it wasn't one of mine. But the good news is that as confusing as immunology has been over the last 100 years, it's really coming into focus, and it's not an exaggeration to say that it's coming of age, because there's been tremendous advances in understanding the different cell types, of which there are many, the different molecules, of which there are hundreds, and how at least some of that fits together. So we know the playlist, we know some of the basic principles of how both the adaptive and innate immune system works, and I'll go over that just a little bit. So it really is quite understandable. You know, I remember someone trying to explain some of the more obscure aspects of immunology when I was a graduate student, and they had to explain several times because I just wasn't getting it. And now that same principle, you can just show a picture of a MHC structure, and you know immediately what's going on. So anyway, things as fields mature, it does get better. I'd also say that on the theme of precision health, which Dean Miner has championed here, and I'm sure he will mention it later today, immunology is almost a poster child for that, because if your immune system isn't doing well, you will not be doing well. I think it's almost a given. And so it behooves us, especially as we move into a new era of medicine, to be paying more and more attention to the immune system and what it tries to tell us about diseases that could happen, that you could be at risk for, or maybe already have, but it's in a subclinical state. And so I think we have a lot to learn there still, especially as we move. I mentioned, you know, I subtitled this "Standing on the Shoulders of Mice" because for about the last 50 years, most of the attention in basic immunology has focused on mouse models of immune responses and vaccination responses and diseases, but those have only gotten us so far. That what you'll hear today from many of the other speakers and myself is how we're moving beyond that knowledge base focused on mouse diseases and mouse immunology into dealing directly with human immunology, human diseases. And we're going to learn a tremendous amount about immunology because it's, you know, most of us don't spend our lives in the basement covered in a HEPA filter the way mice do, nor are we inbred. I'm hoping most of you are not inbred. So there's a whole world here that I think is going to be just fascinating, and we've already seen signs of that. If I had, oh yeah, I can see it here. So this is a short list of all the diseases that are affected by the immune system. And I'm sorry, Sam, I left out organ transplant, but obviously somewhere in there, read in there, organ transplant. But the list just goes on. The more we know, the more we learn that there's tremendous involvement in cardiovascular diseases. Cancer is exploding. Cancer immunology is exploding. You know, it used to be your main options in cancer were chemotherapy and radiation, and there's still a heavy presence there. But more and more, we're seeing miraculous treatments about mobilizing the immune system. It turns out the immune system can recognize cancerous cells, but how to get them to do that, we're just on the cusp of tremendous activities there. It's gone from 100 years of failure to, well, actually, this is now the most exciting thing to happen to oncology in about 100 years, probably. So almost untouched are cardiovascular diseases. We know that's inflammation. We know there are lymphocytes involved, but it just hasn't received a lot of attention. You'll hear about aging. Jörg Ronchi and Connie Wyand are experts, world experts in the field of immune aging. Bali Palendrum, who will talk later, is an expert in vaccination. And the vaccination failures are not his fault. They're the reason he's looking at it, because there have been so many recent vaccine failures that it's become clear that we just don't know enough about the immune response to even something stupid like a flu vaccine. and because the knowledge just wasn't being collected there, and the very specific human knowledge that we need to get. And so some of us, including my own lab, and Bali's have been working just the flu vaccine angle, just to say, well, what actually is happening there? So, and you'll also hear our guest speaker, Linda Lau, will talk about some of the latest advances in cancer immunotherapy in one of the breakout sessions. So moving right along, it's obvious the public wants immunology, that they know they have an immune system, they know it can go south on them, and they're quite interested in how to avoid something bad happening. And this is just one of many products that I see around. You can go to drugstores, you see whole shelves of immune booster. I recently bought a package of dog treats labeled immunity. It goes on, and it's totally fake. There's nothing at all to this because there's no standards. We don't know, and that's one of our objectives, is to define health in immunity. What is immunological health? Well, you know, if you say, what is cardiovascular health, you've got all kinds of lipid assays that you can bring to bear that are standard and help people lead healthier lives. If you go into a clinic and say, how's my immune system? People will look at you like you're some kind of nut, you know. So yeah, we had that course, but no, we can't do anything for you, and we can't look at anything. But now we can measure so many things. It is now really possible to define immunological health, and that's one of our objectives here. And so that people don't have to go to a health food store to figure something out about their immune system, even it's completely misinformation. So this slide I made all by myself, which you can see the technology that went into this. You grab a picture of a cloud from the internet and you make a little black box. So we now know that all medical knowledge is in the form of a cloud, and that but if you ask where's the immune system in that, it's a bit dark. And why is that? Well, because only a few assays have made it into basic medical practice, as I'm sure many of you are well aware, that white blood cell counts, very popular, but that's 1915, and complete blood cell counts, which is kind of a glorified white blood cell count, came in as recently as 1959. So it's as if modern immunology never happened. And there's something like 12 Nobel Prizes from about 1960 on to the present day. There are about 12 Nobel Prizes in immunology. A lot has happened in immunology at the basic science level. It's just not getting over the fence enough into basic medicine. And so I think it's really time to do that and time to tackle that issue. But luckily, we have learned a tremendous amount about the immune system from mouse models and some relationship to humans. And it's starting to make sense. That is, we have a bunch of the white blood cell counts, and not just a white blood cell count or 5 white blood cell counts. There may be 100 or hundreds of white blood cell types fall into 6 or 7 major categories, and they clearly do different things. So things like T cells and B cells are the, doesn't seem like that's really working. That was neat. So T cells and B cells, I'll go into that. We call this the adaptive immune response. These are highly diverse, billions and billions of possible specificities, and it's basically our way of escaping from the evolutionary dynamic that says, well, this virus can replicate every 20 minutes, but we can only replicate every 20 years. So who's going to win the evolutionary race? Well, it's got to go to the virus. So how can we combat that? Well, what we do is we make billions and billions of possible antibodies and T cell receptors, and then have them sitting around on different cells, and then sort of pre-positioning the diversity. All the possible diversity we might need is sort of pre-positioned, sitting there in your white blood cell counts, your white blood cells, your lymphocytes, and they're circulating around, constantly checking for things that don't belong there. And if they find something, they will then get triggered and proliferate and make many more cells of just that type. The other aspect is our cells, what's called the innate immune system. That's on the left-hand side, and Bali will talk a lot about that. But that's sort of your rapid response team. And they have less sophisticated receptors but can nonetheless see things that are characteristic of bacteria, like flagella, like a particular kind of nucleotide substitutions. They're geared to be there everywhere a bacteria or a virus might come in and then trigger a response, start inflammation going, because it's going to be days before the B cells and T cells really get up to speed to make that response. That also, but you'll hear more about that in the other talks. In the adaptive immunity side, one thing I would just want to make clear is that you have this sea of billions of different B cells making antibodies or T cells making T cell receptors, and when they get triggered, a particular one will be picked out by a virus because it binds to that antibody or T cell receptor that triggers that cell to proliferate. So then, because the DNA is already fixed for that particular sequence, all the daughter cells of that the one that gets triggered will be the same. And it actually acts as a barcode. And we're going to be using that extensively in some of the later aspects of what I'll talk about, particularly with T cells. So we call that clonal expansion. Clonal selection goes back to Burnett. McFarland Burnett is in the 1950s postulated this. And it was amazing because at that time, probably one of the most important thought experiments in biology, that he postulated this was happening before we knew lymphocytes made antibodies. He was saying there is a cell making antibodies, and this is the way it works, as opposed to people like Linus Pauling, who thought, okay, the antibody binds, folds itself around the antigen. So one of the great discoveries or predictions in immunology that turned out to be true. All right. Now here's a subtlety, which is the T cell receptors on T cells are quite unique in the sense that while they look like antibodies, they rearrange like antibody genes rearrange and create a tremendous amount of diversity. They're not seeing chunks of antigen the way antibodies are. They're seeing little peptides. They're degradation products and stuck into major histocompatibility antigens. So as you can see here, where it says TCR versus peptide MHC, that's the central event in T cell recognition. And the importance of that is that pathogens can manipulate their structure to avoid antibody detection. Some of the cleverest viruses, for instance, hide the most important bits inside fluffy stuff that they don't care about, but it makes it very difficult to make just the right antibody to neutralize that virus and get protected. Whereas T cells use a completely different system, which depends on degradation and the fact that all proteins, virus or whatever, get degraded into peptides, and then from peptides into amino acids, and they cycle through. What the T cell system does is it uses these MHC molecules to capture these bits of protein, these peptides, 9 or 10 amino acids long, and then go to the cell surface and present them. So you're like the people in Hollywood who go through the garbage cans of the stars and are fishing out like cute little bits and notes and receipts, contracts, agreements, so forth, payoffs, and then they display that to the tabloid press. And in the same way, you're taking advantage of just what every protein has to do, which is it degrades into peptides, and you're capturing just enough information. A 9 or 10 amino acid peptide actually has a tremendous amount of unique information that a T-cells can tell whether that's a peptide that belongs in the cell or whether it's something new that's coming in and indicating something bad is happening. Or it's a cancerous cell and there's something wrong about this cell because its metabolism is all messed up and it's presenting, as we call it something odd. That's what I really like. This is actually called antigen processing and presentation, which back when no one understood really what this was, I love the use of the clinical term that one of the first I learned, which is the patient came in and presented with green foam about the nostrils. And I just thought, yeah, that's you're not saying you know anything about this. You're just saying, this is what walked in the door. This is where the story starts. This is what I see. And now let's try to figure out what's happening. And so it took about 20 years to figure this out, but it's kind of our double helix because it was quite difficult to figure out, but it ended up with a beautiful structure. You can see on the left here on the top is the T cell receptor in blue, and then the MHC in red, and then a little peptide stuck in the MHC, just waiting to be seen by a T cell. The other thing to know, implied but not obvious, was just how diverse these things are. I mean, this rearrangement of gene pieces. And there's some numbers here. It's not incredibly important to know what they are, but they're very large numbers that you start mixing and matching these different bits of antibodies or T cell receptors. You get into the billions or tens of billions or hundreds of billions of possibilities. And that's what saves us. That's what keeps us ready for anything. If something came from Mars tomorrow that was a protein-based life form, you already have T cells and antibodies against that. Rest assured, don't worry. Don't worry. It's, you might have to worry about whose proteins those are, but you definitely have antibodies to them. Don't worry about that. So this is just a cute video. We were involved a lot in trying to understand the mechanics and the molecular biology of T cell recognition using video microscopy. And I remember telling people, we got to get in the movie business. They have more exciting parties than scientists do. And in California, there's a clear path to being governor. So this is a T cell in green attacking a cancer cell. And it's just really spectacular. The little red things are cytotoxic granules. So it's basically just beating up on this cancer cell and killing it using those cytotoxic granules that they touch the cell, and the cell starts just disintegrating. So when that works, it works really well. And this is a prime thing that people are trying to encourage in a lot of the cancer immunotherapy these days, is to motivate those T cells to kill the cancerous cells. And we don't really still know exactly how that's going to work or how to get that to work all the time, but we have things which get it to work, and it's a really active area here, and I bet you'll be hearing more about that from Linda in the next session. So what are we doing here? I think there's basically, we have seen people, especially in this neighborhood, we've seen so many people reinvent whole industries and disrupt industries and do something completely different if they can see it's not working. And I think it's pretty clear that mouse models of disease have taught us a lot, but not enough, that there's not often a very clear path to humans, to translation. We keep hoping there would be. Some of the cancer-related things definitely have come from mouse models, but most things have not. For instance, in type 1 diabetes, the major mouse model, we have cured or prevented type 1 diabetes in that mouse model, I'm told, over 300 times, and we have not cured a single person. Similarly with the mouse model of MS, hundreds of times, hundreds of ways we can stop that disease in the mouse model, but not some of it's carried over to humans, but nothing. There's no cures. So almost has become a remark that a friend of mine, Pedro Lowenstein, introducing a medical school class to immuno-oncology at the time, said, for the mice in the audience, I have wonderful news. Humans, not so much. So how can we turn this around? Well, I think we need new strategies. I think the strategy of just waiting for something to happen in mice and then trying to use that in humans is not working very well. And it suggests to an experimentalist, I think a good experimentalist, you have to listen to the data. You can't just go charging off and saying, well, this is what I believe and this is what I'm going to see. That doesn't work. Biology is too complex. You've got to let the experiments tell you what the right path is. And if you fail, that's just an educational opportunity. You just keep trying and keep failing until something works. So I thought some time ago that what we really needed to do, and Bali has been fantastic at this, is to try some different approach, namely a systems biology approach, which means that collect a lot of data about systems in motion. What is the resting state of something like the immune system. I mean, we call it immune system. We don't typically work on it as a system. But we have the technology now to measure hundreds of cytokines, hundreds of cell types at once, and then see how they change with, say, vaccination or with some sort of intervention, surgery, and so forth. And this will create a whole new way of looking at the immune system. This isn't commonly done in mice at all, but it should be. And it plays on the relatively cell-autonomous aspect of the immune system. The immune system is a collection of specialized cells that talks to each other with cytokines. It's quite simple. I mean, it's not nearly as complicated as a nervous system. And it's all about defense. You know, almost everything you see in the immune system, you can relate to defense or pulling back from say, a situation of self-reactivity or autoimmunity. And so we, with the help of some very generous grants from the NIH and now more recently the Bill and Melinda Gates Foundation and the Sean Parker Foundation, we have set up whole networks of interactions between key clinics here at Stanford with investigators and specializing in many different areas, T cells, B cells, innate immunity, genomics, bioinformatics, microbiome. But a key component of this has been the immune monitoring core. And this came about because in setting up this institute, I thought, you know, what would be transformative potentially in terms of the different components, immunologists and microbiologists and transplant people. And that is a way in which they can get data about the human immune system easily and work with it and discover things. And this has turned into now 11 years on this human immune monitoring core run by Professor Holden Maker. And it has about 25 FTEs and has just been a phenomenal resource for getting immunological data, for storing cells that you really want to have online, and doing many projects, helping people do the immunology of whatever their specialty is. So we have 250 participating investigators. There's been 800 projects, maybe 100 going on at the present time. There are about 25 or 30 individuals in this facility. So it's just been absolutely tremendous in terms of as a resource and as a way to sort of standardize information about the immune system. It allows people like me to do big studies with collaboration of collaborators like Corey Decker and others. They recruit patients, they vaccinate them, we get blood. The blood goes to the immune monitoring core. We get tons of data. We store samples for future experiments, and then we get to work in terms of, well, what does this data mean? What is it trying to tell us? And so this, no one else has anything as sophisticated as this. They're starting to copy it, but I've seen some of the examples, and I would say they're kind of weak, and we're still doing very well. We also, not only are we using the latest technology, but it keeps being the latest because we keep developing technology that we then can put into the center. Gary Nolan discovered this whole technology for mass cytometry, which is like a next generation FACS, fluorescence analysis for 45 markers. And not only did he do a lot with his lab, but he could only do so much, and he basically told us, we got some of these machines into the immune monitoring core, and then suddenly we were, you know, years ahead of what anyone else could do, and funded, enabled a lot of studies. Catherine Blish particularly has done some great stuff with this thing, technology called CyTOF. So we also have software. We'd like to. One advantage of having a standardized facility generating data is that you can then put all of that data in a uniform database and then treat it, you know, do meta-analysis on as more and more studies accumulate. And also I was very proud of, my contribution was our motto, immunology for the people. And somebody asked me if I was from Berkeley. So there's just been a whole lot of stuff has has happened over this 11 years, and we intend that this more and more is going to happen. We've been pouring out papers in the literature. We I think, are really recognized as the world leader in both developing immune monitoring technology for human beings and finding really interesting biology. And I think that's one thing I was realizing as someone who has spent quite a bit of time in my own career developing technology to ask specific questions about basic immunology. I could see that in human immunology, there weren't people like this, that they were all working on mice. What was a problem in mice, people were all over it, but there weren't really people working on the human frontier to say, how can I do this? I can't do this, I can't do what we do in mice. You can't do most of what you do in mice. So we've got to invent new things that will allow us to get high quality information from human beings, both healthy and with specific diseases that we want to know something about. And this is one of the studies that I'll return to in a bit, but it's just one of the cohorts. You know, partly with humans, you want to set up interesting cohorts that are going to tell you more than, say, one thing. And so we got a little bit of money from the Ellison Foundation some years back, 2006 or 7. And also Gary Bathman was able to raise some money and get the whole immune monitoring core going. And then the first thing we did was set up a study where we looked at flu vaccination and then did all these assays based on, you know, people came in, they gave blood, they got a flu shot, they came back, got some more blood, and so forth. And we can analyze the before and after. And this has led to quite a flood of papers, but really, really valuable is a longitudinal study. Nobody does longitudinal studies. They're very expensive. And you want a study that'll go on for 10 or 20 years. How are you going to fund that on a 5-year grant cycle? But luckily, we've been able to cobble together one grant after the other. And this is now headed towards 15 years. So we're doing good. And it's really important because humans are quite diverse. And older humans are more diverse than younger humans. And so to be able to follow the same person from their 50s to 60s, and 70s, and 80s, 90s here has been incredibly valuable. The other way we've used cohorts is something that people had sort of forgotten to do, which is twin studies. If you really want to understand genetics of something like the immune system variation, you really want to look at twins, because here you can have twins that are almost identical in terms of their genome, and then you can just see how, even with that, how do they respond to the environment? What does the environment do? And people weren't doing this, and they were just going around saying, well, the genome will tell us everything about disease. Well, I can tell you that the genome doesn't tell you everything about disease. The genome is what you start with, right? And then life happens, and you live in certain places and are exposed to all sorts of things. And that's where we really want to use twins, because we know that they start in the same place in many cases, but in the same family. We were hoping some had been separated at birth. It turns out that's really doesn't really happen. We're thinking of starting a program like that, though. But I think it's hung up in the IRB for some reason. I don't understand. Such a good experiment. But anyway, we had all this technology, and luckily SRI up the street has the largest twin registry in California. Really has 4,000 twins in the registry, and we're able to recruit with an agreement with them. We've been able to recruit hundreds and hundreds of those twins and give them flu vaccines, measure their immune variables. And so we ended up this paper that came out a few years ago. We were able to analyze 210 twins, age 8 to 82, over a 3-year period, using mostly data from the immune monitoring core. We measured 200 reliable variables of all different kinds, and basically what we found was the environment is most of what drives immune variation. There are some genetic things, but 75% of these 200 variables had an overwhelming non-heritable influence. So the idea that genetics is everything is just an idea and a hope, and a dramatically oversold concept. The environment matters a lot. what you get when you roll around in the dirt as a child is probably really pretty critical. And I say let kids eat dirt. I think they want to. They want to sample their environment. We know that those bugs, some of them are good for you, I suppose. But anyway, so long and short of this is that overwhelming, even young twins, they are more similar than older twins, but they're still very, very different. And so we think the environment really is playing a huge role. Even if they're living in the same household, the environment must be playing an enormous role. And one thing we stumbled over here that was surprising was the influence of cytomegalovirus. Now, as some of you know, cytomegalovirus is a herpes virus that is ubiquitous around the world. In the US, something like 50% of the people are infected with cytomegalovirus. It doesn't seem to harm them in most cases. Around the world, billions of people are infected with this virus. And we had a unique opportunity here because it turned out that 16 pairs of our monozygotic twins in this study were discordant for cytomegalovirus. Something you can assay very simply in the blood sample, see if you have antibodies, and half these people did and half didn't. So we could look at people with identical genetic backgrounds and CMV plus and minus, and turned out there were massive changes in the immune system. That cytomegalovirus completely reshapes a lot of this, the immune variables, almost 60%, as it says here. And that was really striking, and says a little scary to say how this one virus could just come in and start remodeling your whole system. But what is the actual effect? Well, that came in from another study that we were doing independently on this Stanford-Ellison cohort, and we were following up on some ideas people had put forward that the cytomegalovirus infection could accelerate aging, that because they saw increased mortality of very older people in a Swedish study that were cytomegalovirus positive. But and so we looked at our older individuals and our younger individuals, and on the right are the older individuals. So we basically couldn't see really much of a difference between cytomegalovirus positive and negative. But in the younger cohort, we had about 1/3 were in their 20s, perfect in every way. And actually there, you look at the yellow on the left, those are the cytomegalovirus positive young people. They had a better flu vaccine response than cytomegalovirus negative younger people. So that says that CMV is actually boosting your immune system. And we saw data in mice that were exactly in line with that it almost rescued the mice. If they'd been infected previously with the murine CMV, they did much better against the flu infection. So cytomegalovirus, we think of it as a pathogen, and if somebody has just irradiated you, it is a pathogen. It can kill you, and does kill you if your immune system is severely disabled. But for healthy people, it actually seems to be a benefit, and that could be why it's so ubiquitous around the world. So that was really one of the surprising things. Another thing is, I mentioned cardiovascular data and the influence of the immune system on cardiovascular health. And here we found work of David Furman has been really good here, collaborating with some of the great cardiologists we have here, like Francois Haddad and Joe Wu. He had a paper just last year where he found in this Ellison cohort people with chronic hypertension, and then other people that did not have hypertension. And he found all kinds of sequelae associated with chronic hypertension, including stiffening arteries. Things were not looking so good. And a long story short was he found in the study, he found not only there was an inflammasome, so inflammasome is one of the immune system inflammation pathways associated with infection, but here people with hypertension had this inflammasome high phenotype. So the inflammasome chronic inflammation was working, and you'll see that actually both Jörg Ronchi and Connie Wyan, I think, are going to touch on that topic in later, this inflamma-aging concept. But anyway, we're seeing clear evidence of a link between a particular immune inflammation pathway and hypertension. And furthermore, we found upstream of that was a nucleotide metabolism defect, and we could identify some metabolites that stimulated an inflammasome pathway by themselves that were novel. And also, if we put them in mice, they gave the mice hypertension. So it's kind of a reverse translation experiment, I know, but it's a proof of principle that these things we're finding really are important. But I would say the anecdote to this, it turns out that coffee and dark chocolate suppress inflammasome activity. So any of you are not drinking coffee or eating dark chocolate, get on that right away. It's definitely, it's got to help. It's got to help, yeah. I just want to also mention that we've analyzed so many people now. We have over 1,000 relatively healthy people that we've analyzed using the full suite of the immune monitoring core. And with the help of David Furman and Jose Montoya and Francois Haddad, we have this 1,000 immunome study where we've been able to just preliminarily identify people that are aging in a healthy way versus people that are not. And we can see a subset of people that are doing extremely well, even in their 80s, other people not doing well based on chronic inflammation and so forth. So we're just working on this paper, and hopefully we'll have it out soon. But it goes along with our hope that we'll be able to identify robust markers of whether your immune system is doing well or not, metrics of immunological health, or as someone said, oh, you want to make an immunometer. Yeah, we do. And so I think I'm going to have to wrap up pretty soon here. I'm seeing a red light and so forth. That there's still a lot to talk about. But anyway, we will go through this quickly. We do see some dramatic data in humans that has changed our picture of immunology in general. I mean, there's a prejudice in the field that you're only going to learn basic immunology from mice, and that humans are about translation. And I think it definitely is about translation, but I also think because it's such a different system and because there's a full onset of genetic heterogeneity, environmental heterogeneity, we should, if we're not learning something new about the immune system, we're not doing our job. And so one technology we developed more than 20 years ago involves a method for multimerizing specific peptide MHCs and labeling T cells because of that interaction. And this allowed us to take a new look at an old concept in immunology, which in this, everything goes back to Burnett, basically. But this is where Burnett was wrong. Burnett in '59 proposed that cell-specific T cell lymphocytes were deleted to avoid autoimmunity and deleted during development. And this was apparently confirmed. You know, if you took an immunology class sometime in the last 25 years, you would have heard this as canon law, that we looked at this in the thymus, massive cell death for T cells that had self-specificity. And that's the end of it, and no one considered that they had to do anything further. But by accident, we were just going to the blood bank and looking at random people for particular viral responses that they hadn't had yet, like HIV, like bird flu, like HCV. And we saw a certain frequency of T cells using this T cell labeling technique. And then we decided, okay, those are the foreign peptides. Of course, they're going to be more frequent. What about self-peptides? They should be much lower frequency because they're self-reactive and they're going to be deleted. And then you look at that on the right-hand side, you don't see any difference really in terms of frequency. So what happens to deletion? That seems to be so obvious. Well, we looked at this in more detail by looking at a Y chromosome encoded T cell antigen called HY. And we did see a difference. Men had a lower frequency than women because it's a self-antigen in men and a foreign antigen in women. But it wasn't a big difference. It was about a 2- or 3-fold difference. And mice were even less. So it wasn't as big a factor as people had thought. And why is that? Well, the explanation, I think, is that tolerance works, is that autoimmunity is not a big deal in children and young adults. Less than 1%. It's more a disease of older people, middle and older people. All evolution cares about, I'm sorry to say, is children and young adults. And the biggest threat to children and young adults back in the day before vaccination and basic hygiene was infectious diseases. That infectious diseases killed something like 50% of children under 5, generation after generation, and a lot of young adults as well. So that's by far a much more significant force in evolution. So if you're an immune system, you've got to be focused on infectious diseases, got to be focused on having as complete a repertoire as possible, because you don't know what's going to come after you. And autoimmunity is not even a rounding error. So anyway, so this is something very basic, and the immune system, the field is still kind of coming to grips with how could this be true? We believed this other thing for so long, but that's okay. They'll get there. And then the last topic really has to do with T cells, a specialty in the way that we've worried about for a long time, and about the technologies that we've used to develop and get to the heart of T cell responses in diseases. And the shocking thing is we really haven't had a good grip on T cells in autoimmunity or in cancer without these tools. And so this happens to be one. We had single cell sequencing of T cell receptors. I won't go into the details, but I will take you back to what I mentioned about clonal expansion, is that with the single cell sequencing assay, you basically, if you see, like in red here, you see that the same T cell receptor sequence in 60 different cells that you pulled out of a tumor, in this case colon carcinoma, that means something. Those all came from the same original cell. It got into that tumor, it liked something that it saw, it proliferated. It made many daughter cells and generations. And in fact, when we look in tumors like this colon carcinoma, we see that more than half of the cells have divided and produced more than one cell with that exact same T cell receptor. That's what I mean by using a barcode. Whereas the adjacent tissue, the colon carcinoma, the adjacent normal tissue, you don't really see a lot of clonal expansion. It's very diverse, like most things. And our colleague Chris Garcia developed a nice technique of a way of finding T cell peptides for particular T cells. I'm going to have to go through this really quickly. And he's applied this to the colon carcinoma area, and we found several peptides, several antigens that these T cells see. One is a so-called neoantigen, which is a result of a mutation in the gene in the tumor that the T cell is recognizing. And there's a lot of excitement about those. Another one is shared between multiple patients, and so that's going on. We've also developed software that allows us to zero in on what is important in terms of T cell specificity directly from sequence information. The software is called Glyph, and it has allowed us to group T cell receptor sequences that we really know nothing about except the actual sequences, and we can group them into clusters of T cells that see exactly the same peptide in MHC, and we could show that in this particular study of TB-infected people that we published recently. And this means that we now can have a much better handle on taking advantage of the technology that we have to get lots and lots of T cell receptor sequences. And this is sort of is a summary statement here where by looking at these things, we can see these clonal expansions using the T cell receptor sequence as a barcode. And this shows us lots of unusual activity in many diseases directly just from blood. What can you do in humans? You can get blood, really easily. So this is an example. These are patients with multiple sclerosis. And top line are healthy controls, and we're looking at particular T cells there. And we see very little clonal expansion. If you see all yellow, that means every sequence of hundreds is unique. But then you see a few that are expanded, but in very low level. But in the MS patients, we see massive expansions of CD8 T cells. That says there's something going on there. That says those T cells are involved in this disease somehow. And with our other technology, we can now go and find out what are the antigens, what are those T cells seeing, and what is this going to tell us about the disease? Are they seeing a pathogen? Are they seeing self molecules? It's really not well known in MS or any autoimmune disease, and we now have a way to do that. Furthermore, we see this everywhere. Here's just a summary where, again, we saw the healthy, but if you look at Lyme, people with a Lyme infection, you see this clonal expansion. You look at MS on the bottom, you see that. But in chronic fatigue, which nobody knows anything about, it's clearly there's an immunological component because we're seeing much more than the normal level of clonal expansion. And again, we can now reach in, we can take those T cell receptors and find out what they recognize. And that will tell us something important about the disease, about and impact both diagnosis and, I'm sure, treatment. So, okay, we're at the conclusion. So, so my conclusion is that this field is exploding. We're finally able to move on from a lot of interesting, important, scientifically amazing work in mouse immunology, but that's really just the bedrock on which we really want to get to humans. And we're starting to be able to get to humans. It requires new technology, new thinking, new strategies, but it's already having a huge impact in cancer, and I think it's going to have a huge impact in virtually every other area of medicine. So I think it's well worth keeping an eye on for what's going to happen, because I think a lot is going to happen, and I think you should be proud of being here that we're really leading the way in this kind of really new leap forward in immunology. You know, at the end, you know, we hope to have develop these metrics of immunological health that could be a benchmark, just the way cholesterol tests are a benchmark for cardiovascular health. Imperfect, but still the gold standard. We hope to do that with the immune system. So I should stop here and still have time for some questions. So.
Thank you very much for your talk. Do you have any idea in terms of the T cells, whether they're recognized in the actual sequence of the peptides, or is it a three-dimensional structure that's still maintained by the peptides?
No.
And how do you conceptualize what the T cell actually sees?
Sure. Well, we don't have to conceptualize too much because we have many structures that position the T cell receptor over the peptide MHC at molecular resolution. So a very important point is that the pathogen cannot manipulate the structure of its peptide. It gets degraded just like every protein does, and that already releases it from most of whatever structure it had in the original protein. And then even more so, the MHC is like a bear trap. It's just closing around the peptide and forces it into the structure that suits the MHC. It's a 600-pound gorilla, you know, grabbing a banana. So the only way the pathogen can manipulate the T cell response is by mutation. And that's how HIV can be so deadly. But most pathogens can't do that in real time, or not nearly as efficiently as they might need. So yeah. Does that answer your question? Yeah. All right. Yes?
Yeah, speaking of immune booster products,
Yeah.
are there any for which there is,
I have some here.
in your view, some reasonable evidence for efficacy?
You know, I don't think they bother. You know, I haven't seen any studies.
They don't bother?
I don't think they bother to do anything, yeah. I mean, they might read, you know, zinc. Somebody will say zinc deficiency leads to immune deficiency, which is true. And then they'll put zinc in something. And they'll say, okay, this will help your immune system because it has zinc in it. I mean, they don't have, there's no, even if they did do clinical studies, they have no benchmarks. And we also know, you know, too much of immune boosting can lead to autoimmunity. In fact, that happens in some of the cancer immunotherapies. They boost up the immune system so much that you get autoimmunity. Not good. So we need a much more sophisticated understanding of what would be appropriate for actually probably an individual in terms of just how much to boost and with what, and then what are we aiming for. So it's a complex issue that I don't see any people producing these products. They're just out to make a buck. They're not serious, except about making a buck. No.
Thanks.
Nope, sorry. You know, I think that could be the downside, but I think in the, you know, again, in an evolutionary context, autoimmunity is such a, you know, probably partly a disease of civilization and hygiene as much as anything. You know, how much of it was a factor back in the day when, you know, you lived to be 30 or 40. So I don't think, I think, you know, I do subscribe to the belief that autoimmunity is generally caused by a pathogen trigger, because we know that that can break the sort of tolerance mechanisms that we have against self-reactivity. So I still think that that's the best candidate, but there really aren't many examples of that. In fact, almost no examples of that in humans especially. So that's where the T cell work will help, because we'll see whether these T cells are specific for a pathogen or a self antigen or both, which, you know, would be part of the thing. Yeah. Yes. Okay.
Given your explanation of the gorilla with the banana, and thinking about flu, which is much more common, and what happened in 1918, what's your thoughts, or what's going on about how that might change? It's in the newspapers now, so there must be some science.
Right. Well, it's a huge area of discussion right now. And unfortunately, what happened in the vaccine business was that it just got very lazy, and people were satisfied with a flu vaccine that is only protective for maybe 30% of older people on a good year, and maybe 10% in a bad year. And just like they weren't making a lot of money, and so they didn't spend a lot of time trying to improve things. So it's really good that the NIH and I also saw recently the Gates Foundation is starting to say, well, look, we now know enough that it's really achievable to get a universal flu vaccine off the ground. We think we know enough to make that possible. We don't know the details, and of course, the devil tends to be the details in terms of all the things that go wrong with the vaccine. But I think we're on a path where there's enough scientific input, structural input, and infectious disease and other inputs that really should make a go of it and come up with something much better than what we have, which is, you know, hasn't changed in 50 years and is pretty stupid vaccine. In fact, it's actually good. That's why one reason we use it for two reasons. One is everyone can get it. It's not restricted like most vaccines. But two is it's a crappy vaccine. So you have a lot of non-responders. So you don't have to do thousands of people to see a few non-responders. You see 24 people, you get a dozen non-responders. So really good for the science aspect of it. But crappy vaccine. Really, it's an embarrassment that we have a vaccine like this and have had it for so long. And then the most creative ideas are, you know, in that area is, oh, let's double the amount of vaccine or quadruple it. I mean, this is what passes for, you know, innovation. So it deserves to be disrupted and to find really much better things that we can do. And we need to do it because the bad flus are coming, you know, that they're really these deadly, not just the bird flu, which is H5N1, but there's also this flu out of China, which is H1N7, something like that, has a 7 in it. And that's killing people in parts of China. So, and these things, unlike SARS, where you have symptoms before you're infectious, someone has pointed out to me that with flu, you're infectious before you have symptoms. So with international travel, a lot of people are gonna, you know, if a really bad flu comes along, a lot of people are gonna get it before you can even, you know, put controls in place. So So we really need to put a lot of effort into this and get it right.